हेलो एवरीवन, वेलकम टू बायोसाइंट वंस अगेन। आई होप आप सब अच्छे होंगे। टुडे वी आर गोइंग टू स्टडी अ वेरी इंपॉर्टेंट टॉपिक ऑफ क्लास 12 and it is very important for your NEET exam. So let's get started. The name of the chapter is Biotechnology. Try to understand why are we studying this chapter and how is it important in our day-to-day life. I'll be giving you one example that more or less you see people around you. In every home there is a person suffering from diabetes, right? Diabetes is diagnosed as the lack of insulin in the body. We know that from the genetics that insulin is a hormone which is produced by some gene which is already there in your DNA. Now, what about that? You are taking out this defective gene which is not working, and a person is getting same protein from outside. No wonder everyone knows that the insulin injections are in market now, and we can purchase it and we can save many people's life. Now, how scientists did this? So that is all about the biotechnology, where we will be making such products which is going to be beneficial for the human, and it is for the human welfare, where we will be using living organism or their products or their enzymes to produce such kind of products which are good or better for the society. Okay, similarly, we know that any innovation or anything new comes into the market, there are challenges of acceptance by the people. So similarly happened with biotechnology also. Now we have plenty of products which are actually doing good in market, and we are now well established trying new technology in the biology. Okay, let's get started. So the use of natural science and living organism to create products and services, that's called biotechnology. Bio means living, and technology, you know very well how we can use the technology and we can change the lives of many people. Okay, so what are the tools required? See, if we are talking about the technology, definitely we should have certain tools for that, and we have those tools. And how we are gonna use those tools, just after the completion of this lecture, you will be surely very positive that now we understood a topic. So here are 5 tools which are required by any biotechnologist or genetic engineer. The first one is called enzymes, the second one is called vector, the third one is competent host. Obviously the technology we will be using here, and the whenever we complete this process we need to commercialize it, so it should be, the production of the product should be on commercial level. We will be dealing it one by one about the each tool. First I will be explaining you about the enzymes. Enzymes are like biocatalyst. Enzymes are very important whenever we talk about the biological reaction. Like in chemistry you study about the catalyst, here we talk about the biocatalysts. Okay, so first what do you need if you want to extract something from somewhere? Like if you are extracting something from a plant cell, let's suppose that you have a plant cell here. Now you want to take out its DNA to make certain product. What do you need? You have to go from outside to inside to extract the DNA. Do you know what is the cell wall of plant made up of? You know that it's cellulose. So which enzyme will lyse or break that plant cell wall? The first enzyme you would require is cellulase, isn't it? Cellulase will help you to, other than cellulose, it also have what? Pectin. So you can use pectinase, and then the cell membrane which is made up of protein. So you must be requiring protease, and then also lipid. So you need lipase. Like this, you have reached to the DNA and you can easily extract the plant DNA. Now, if your target is an animal cell, what do you require? Just two things, nothing more than that. What is that? Yes, you are absolutely correct. You just need protease and lipase. Very good. Now, if it is a fungal cell, what do you need now? If it is a fungal cell, what do you need to lyse it? Nothing but chitinase. Because the fungal cell wall is made up of chitin. If it is a bacterial cell, what do you require then? If it is bacterial cell, what do you require? Nothing more than that. It's also present in your saliva. Yes, you are absolutely correct. It's lysozyme which breaks the cell wall of the bacteria. So the first enzyme which you require is the lysing enzyme, and I have told you about the different cells. Okay. Now, here, the another enzyme called endonucleases. Nucleases. We are talking about the nucleic acid. We are talking about the DNA. Okay. So, why is it called endonucleases? Because this is found in some bacteria and which makes or creates a nick or a incision in between the DNA. That is why it is called endonucleases. Let me explain you here how endonucleases works. You know that the DNA runs from 5 prime to 3 prime and because the strand is anti-parallel 3 prime to 5 prime. Now here it would be very important to emphasize on a term called palindrome. I hope you have heard the term palindrome. The palindromes are such words, if you speak it or write it from left to right or right to left, it will be same in pronunciation. Like if you know Malayalam, Malayalam. Isn't it left to right? If you read it, it is same. This is a palindrome. But we are talking about the double-stranded DNA. Now I am telling you that here GAATTC is the one strand and the opposite strand will be GAATTC. Okay. So now you have the two kinds of endonucleases here, and endonucleases, especially the restriction endonucleases creates a nick at a specific nucleotide pair. So if we talk about EcoR1, it creates a nick here between G and A, guanine and adenine. So here you will be getting what? A small piece of DNA and a big piece of DNA like this. So such kind of ends produced by the restriction endonuclease is called as sticky ends. What do we call it? Sticky end. I'll be telling you what are the advantage of using sticky ends. Suppose if the nick is created in between somewhere, this is sticky ends or cos ends. Sticky ends. If we talk about the parallel cut, this may generate what? It may generate the blunt end. Blunt end. Okay. The restriction endonucleases or the endonucleases are named according to the organism out of which it is extracted, and endonucleases helps in the defense mechanism of the bacteria. So here if we talk about EcoR1, I will just help you to understand. You must have seen it somewhere that EcoR1 It is extracted from Escherichia coli. So E is the first alphabet is for the genus name, CO is for the species name, this R is the strain name, and this is the first extracted endonuclease from the E. coli. That's why it is named as EcoR1. We have a very important question asked in many years. It was asked, so you should know the first discovered, the first discovered, first discovered, first, sorry, first isolated, first isolated, first isolated endonuclease is, do you know the answer? Is, because discovered is different and the isolated is different. The first discovered is hind 2. This is the first discovered, first isolated. The first discovered is hind 1. Haemophilus influenza. D is the strain name because it is RD, the strain name, but we have already given this R to EcoR1. So here it would be hind 2. This is the first isolated. This is very, very important. Very, very important question. Okay. So now the next enzyme is what? Alkaline phosphatase. Alkaline phosphatase, you have to understand it here that it is important because whenever we have a piece of DNA, it will always have a P and that is the on the fifth carbon and the OH group on the third carbon. If you are just opening a plasmid to introduce your DNA, it is a high chance that having a sticky end, it will easily form the bond. So you will have problem in the insertion of your desired DNA. So what is the role of alkaline phosphatase is it removes, it removes, it removes phosphate from DNA, from DNA, so that it will prevent the formation of the ring once again, phosphate from the DNA. This is alkaline phosphatase role. Now once your desired DNA is inserted, I forgot to mention it here, endonucleases are also called as molecular seizures. Molecular seizures, molecular seizures, whereas the ligases are called as molecular glue. So I need not to tell you what is the function of glue here. It will be helping you or the scientists to again form the phosphodiester bond and phosphorylase. This ligase is for the sealing the plasmid after insertion, and then phosphorylase will help in the making of the phosphodiester bond once again. That is how the enzymes are important for the biotechnology process. In the coming video, I will be discussing about the vectors, and the most common vector is the plasmid. I'll be discussing about that. Till then, stay tuned. Thank you.